Hello, everyone. This is Tom Roberts. I'm really, truly honored to welcome you to the Emily Frederick DiMaggio Lecture. This seminar is funded by the DiMaggio family, and we're saddened to share that Mrs. Emily Frederick DiMaggio passed away in February of this year. The entire Dana-Farber Cancer Institute extends our deepest sympathies to her family, and we are honored that her children, Emily, Peter, and Paul, are tuned into today's presentation in her memory. The Emily Frederick DiMaggio Lecture is an annual seminar in basic science that was founded in 1978 through a gift made by Emily and Dom DiMaggio. Emily was a longtime trustee, donor, and fundraiser who devoted her remarkable energy and talents to advancing many of Dana-Farber's charitable missions to benefit children, culture, and health. The late Dom DiMaggio was a 7-time All-Star and 10-year veteran of the Boston Red Sox and longtime teammate of Ted Williams. We're forever grateful to the DiMaggio family for their generosity and support, and to Emily for her commitment and exceptional service to advance Dana-Farber's mission over the years. We're deeply honored to hold this seminar, which bears her name. And I am deeply honored to introduce our DiMaggio family seminar speaker, Dr. Peter Vogt. I have to say that if scientists are allowed to have heroes, and Peter is one of my heroes. His work over many years has been so outstanding and has led to so many discoveries. It's just hard to hold it in proper esteem. I always tell my postdocs in particular to look at the very elegance of his genetic studies on oncogenes. They're just amazing. Peter was educated in Germany. He got his Bachelor of Science from the University of Würzburg and his PhD from Tübingen University in Germany. After a postdoc at the University UC Berkeley in the virus laboratory, he moved on to be an assistant and associate professor at the University of Colorado and an associate and full professor at the University of Washington before moving to a much longer tenure at USC School of Medicine in LA. And for the last several decades, more than several decades, he has been a professor and leader at the Scripps Research Institute in La Jolla. We always say that our speakers have many honors. Peter's list of honors is somewhat longer than my CV. I will name a few. The Bristol Myers Award, the Charles S. Mott Prize, General Motors Cancer Research Foundation, the St. George Prize, the Pescalor Foundation AACR International Award for Cancer Institute. He is a member of the National Academy of Sciences, the Institute of Medicine, now the National Academy of Medicine, and the American Academy of Arts and Sciences. The title of Peter's talk is What to Do with an Oncogene, and I assure you there is no person on this planet better suited to give this speech. Peter, welcome.
Thank you, Tom. It was a very kind introduction. And I am really honored to be here and glad to be able to give that seminar. So you might ask yourself, what does it mean what to do with an oncogene. And what oncogene is he talking about? Well, the oncogene I'm talking about is the same gene that Tom Roberts and Lou Cantley many years ago discovered and pioneered. And I want to cover three topics. The first is to look at the effect of the FDA-approved PI3 kinase inhibitor, Bill 719, that also goes under different names, on the cellular signaling proliferation and the transcriptome. Then I will turn to something that's very close to my heart, and that's identification of mutant-specific ligands. And finally, I'll discuss our high-resolution single-particle cryo-EM analysis of 3 functionally distinct conformations of PI3 kinase. For those who don't work with PI3 kinase every day, here's a quick start guide. It's a lipid kinase, and it's a dimeric molecule consisting of a regulatory and a catalytic subunit. And it links receptor tyrosine kinases and G protein coupled receptors to downstream kinases, including AKT and TOR. It's absolutely necessary for the survival of normal cells as well as cancer cells. And there are cancer-specific mutations in PI3 kinase. These mutations have gain of function, increased kinase activity, and are often considered to be independent of upstream signaling, but this isn't quite true. You do see a response of the mutant to upstream signaling. And if we look at inhibitors, there are more than 100 inhibitors that have been disclosed, but only 5 are FDA approved. And of these 5, only 1 is approved for solid tumors. And that's alpelisib or Piqray or Bill 719. I'll use the Bill designation and alpelisib interchangeably. It is approved for breast cancer. It is a P110 alpha isoform specific inhibitor. It's now approved 3 years ago. Official clinical indications are the tumor should be PI3K mutated, hormone receptor positive breast cancer. The effect of the inhibitor in the phase 3 trial was an increased survival from 6 to 11 months. And significant side effects have been observed, notably hyperglycemia. And in the phase 3 trial, just as an illustration of the side effects, about 25% of the patients discontinued treatment because of the side effects. So the questions I want to ask are, what is the effect of the inhibitor on proliferation of mutant and of wild type cells? How does mutant and wild type PI3 kinase signaling respond to an inhibitor? And what can we learn about inhibitor action from RNA-seq? We will use MCF10A, which is a continuous cell line derived from human breast epithelium, and the same cell line with a single allele knock-in of the H1047R mutation, which is the most active mutation and also one of the most frequent mutations in cancer. We will use a serum-free, chemically defined growth medium to exclude any growth factor effects. We will use several variables, the inhibitor versus no inhibitor, the mutant cell line versus wild type cell line, and as growth factors, we will use insulin and EGF versus no growth factors. And we'll analyze cell proliferation, indicators of PI3K signaling, and RNA-seq will be analyzed by principal component analysis, T-distributed stochastic neighbor embedding, and gene set enrichment analysis. Cell proliferation. So we have wild type on the left, mutant on the right. And you can see that the inhibitor does affect and inhibit cell proliferation, and that inhibition is attenuated by growth factors. EGF is much more effective in this attenuation than insulin. And the same is true for the mutant cell line. You get vigorous replication without inhibitor. And if you use inhibitor, the replication is attenuated. Now let's look at signaling, and we use S6 as an indicator. Wild type, insulin increases signaling, EGF does a better job, and both together generate a huge signal. Now, the inhibitor attenuates, but EGF and both growth factors still come through with a very strong signal. And the same is true of the mutant cell line. And in this case, we have used conditions where we see almost no signal without a growth factor, which is not normally seen. If you use this mutant cell line without growth factor, you usually do get a signal. But insulin increases the signal, and so does EGF. And the inhibitor, again, attenuates this increase, but it does not extinguish it. So we have an effect of growth factors on mutant signaling and on wild type signaling. And if we look at the transcriptome with principal component analysis, we get 3 clusters. One cluster corresponds to no growth factor. Second cluster corresponds to insulin present, and the third cluster corresponds to EGF. And now the question arises, what happens if we add inhibitor? So here we have, again, these clusters of transcriptomes, no growth factor, insulin, EGF. We add inhibitor, we get a change in the transcriptome. And the changes are always toward the non-stimulated cell transcriptome, but you never reach the non-stimulated stage. This is true of EGF, it's true of insulin, and even the originally non-stimulated cells which receive inhibitor, there is a slight change in the transcriptome. And if we now look at the mutant cell, we see the same thing. EGF, insulin, and non-stimulated. And with an inhibitor, we get a change in the transcriptome, but we never restore the non-stimulated condition. And TSNE shows the same, essentially the same situation. Here we have no growth factor, insulin, and EGF. And if we add the inhibitor, we never get back to the unstimulated. We move these transcriptomes, but we never can regain the original state. And that's true of the mutant cell line as well. Here we have no inhibitor, unstimulated, insulin, EGF, and we go back and add the inhibitor, and we change the transcriptomes, but we never reach that unstimulated state. Well, let's go to GSEA. And the most interesting signature that we find in the mutant cell line is a signature that is very close to the signature for basal breast cancer. And you have the critical genes for basal breast cancer are up as for basal breast cancer. And then there are some genes that are down. And I want to discuss this GSEA analysis from various angles. First, I want to talk about the effect of the inhibitor on signaling hallmarks. So it's with inhibitor versus without inhibitor. And then I want to discuss the effect of growth factors on these signaling hallmarks. And this will be with growth factor and without growth factor. And then I want to switch. What I'm looking at here is not the entire transcriptome, but the differential between wild type and mutant. And that differential includes effects on signaling hallmarks, and it is reflected in that basal breast cancer signature. So the basal breast cancer signature is a differential signature between wild type and mutant. And let's start with the inhibitor. You can see that MYC targets and TOR targets are all downregulated, as you would expect. So the inhibitor works as advertised. RAS targets are barely affected. Now, if we look at growth factors and the signaling hallmarks, we can also see that the growth factors strongly upregulate MYC targets, strongly upregulate TOR target signaling, and RAS pretty much unaffected. So now what I want to do is look at the mutant differential. So whatever the mutant induces, a differential in MYC targets and a differential in TOR target is not affected by the inhibitor. This is, and it's important to make this clear, that I'm not looking at the entire transcriptome. I'm looking at this difference between wild type and mutant cells. And that difference is not extinguished by the inhibitor. And finally, we look at this breast cancer signature for basal breast cancer. And again, this is a differential, and that differential is not affected by the inhibitor. The genes that are normally up, stay up. The genes that are normally down, stay down. So let's go back to the questions we asked. We found that we do get an effect of inhibition of the inhibitor, but we don't get a no signal state if we add growth factors. Then the mutant specific increases in enrichment scores for MYC targets and TOR targets, as well as the signature for basal breast cancer, are not significantly changed by the inhibitor. And it's clear that this makes it important to understand the molecular basis for clinical benefits of PI3 kinase inhibitors. And that I only have suggestions, and these might be microenvironment, immune regulation, but that question definitely needs additional work. So what would be the next steps for PI3 kinase inhibitors? Because PI3 kinase is essential for cell survival. Usually the therapeutic window is fairly small before significant side effects make it impossible to apply it in the clinic. So we need to target cancer-specific mutations. I mean, that suggestion has been out there for a long, long time, and it's always brushed aside as totally unrealistic. And I want to show you that it is not totally unrealistic. One of the conditions is we need to access a different kind of chemical space. What we do now is really essentially use enzyme inhibitors. We don't work with allosteric inhibitors. We don't look for allosteric inhibitors. So there is a lot of chemical space out there that's unused. And the other important issue is structural information. Most of the structural information we have now comes from X-ray crystallography. We need more. And I would say we should use cryo-EM much more effectively and much more intensively. So let me begin with the other chemical space. This is a collaboration with Dario Neri's group at the ETH in Zurich. And Dario designed a DNA encoded library according to the following principles. There's a scaffold which occurs in two stereoisomers, and that scaffold is linked to one kind of building block that we will call building block A. And this connection is encoded in a DNA sequence that is linked to the scaffold, and we call that sequence code A. Then a second building block is attached to the scaffold, and again, that building block is encoded in a DNA sequence referred to as code B. So we end up with a library where the compound consists of a scaffold, two building blocks, and the code A and code B. We can easily identify the compound from the sequence of A and B. And the way this library is screened against PI3 kinase is as follows. PI3 kinase is purified and it is biotinylated, and then it is immobilized on stepped avidine beads. So we have a solid support for PI3 kinase. Then the whole library, consisting of 366 and so 1,000 compounds, is exposed to that immobilized target. The non-bound compounds are washed off. The bound compounds are recovered by heat denaturation of PI3 kinase. And as a next step, the compounds are sequenced. The DNA is sequenced. It is PCR amplified and then sequenced, and the sequence provides a fingerprint of the frequency with which a certain DNA sequence is bound to the compound. Now, first we did control. This is just the stepped avidine streptavidin surface and exposed to the library. So there's no protein involved. We want to eliminate any non-specific binders. And we found one in the library, and it's the same one, whether we use a single-stranded DNA or double-stranded DNA. With double-stranded DNA, the background is clearly much better than single-stranded DNA because of the stiffness of the double strand. There's much fewer non-specific interaction. Then we checked wild type. And as you can see, we do find some very strong binders in the sequencing. And the strength of these binders was confirmed in a special ELISA assay. So it's in the nanomolar range. So we have specific binders for the wild type. And then we checked mutant binders. And again, we find some very high affinity binders. And these binders, again, were validated in the ELISA assay in the nanomolar range. So if we compare, we find a wild type specific binder that's very, very high affinity. And we find 3 mutant specific binders. And the mutant is the 1047, H1047R mutation. So that's essentially where the experiments are now. Where do we go from here? So there are 2 possibilities. One would be, why don't you use the mutant-specific binders to make PROTACs? And that's an option, but it takes a lot of experimental work that we have still to do, determining cell penetration and binding to PI3 kinase in the cell, and then constructing the PROTAC. So, but it is a realistic way to proceed. The other way would be for a skillful medicinal chemist to look at these ligands and build inhibitors. Obviously, these inhibitors would have to be allosteric inhibitors. The ligands by themselves do not inhibit PI3 kinase. They bind, but do not inhibit. So there are 2 ways to go. One is to use them to build inhibitors, and the other is to build PROTACs. So now let me turn to single particle cryo-EM analysis of the P110 alpha P85 alpha dimer. I want to discuss 3 conformations: unliganded, Bill 719 bound, and phosphopeptide activated. Let's begin with the unliganded. Here you have the 2D average analysis, and this is the electron density map. This is the model, and this is or reflects the resolution. So we are between 2 and 3 and sometimes 4 angstrom resolution. We can identify individual domains. We can identify the ABD. Unfortunately, there is something here. Here we go. Here's the ABD. We can identify the kinase domain, and we'll come back to this. We can identify the helical domain. It's over here. The C2 domain is barely visible here. And the RAS-bind domain is up here. If we look at P85, we're not doing as well. We identify the ISH2 domain. We identify the NSH2 domain, which is that pink one over here. And that's about it. We don't identify SH3, BH, and the CSH2 domain. There is something that is not, these are too movable. They move around too much in the unliganted PI3 kinase. And so they are not captured in an image. Then we looked at the BIL 719 bound structure. And the first thing we saw is there is some extra density. It's not high resolution, but it gives us a shape that we had not seen before in the unliganted structure. And it immediately raises the question, can we identify this extra density with the missing domains of P85? The answer is, by now we have a good idea what is what, but originally we just saw these three domains and we referred to it as ED1, ED2, and ED3. In the paper that we published, we guessed what is what, and we guessed wrong. So in the meantime, we have more definitive data, and we can now say that this is the SH3. Oops, this is SH3. And this is a CSH2, and this is a BH domain. It's important because binding to the inhibitor induces a conformational change in the molecule. And that conformational change is of immense functional consequence. If you look at it, you can see that the CSH2 domain now blocks access to the kinase domain, to the active site of P110. So not only is that an ATP competitive inhibitor, it is also a small molecule that induces a conformational change. So it would be an allosteric inhibitor as well by blocking access to the active site. We have high resolution analysis. And what you see here, all the colored areas are contact regions with a CSH2. And you see those in the C lobe and N lobe of the kinase domain. The yellow molecule here is BIL 719. There's also contact with ISH2. So that was a big surprise. Here's a small molecule ATP competitive inhibitor, and it binds and uses a conformational change that really amounts to also acting as an allosteric inhibitor. Then activated PI3 kinase, and that's the most difficult and most troublesome of this whole part. We thought it would be easy, but the only 2D class average that we can identify is the core of the molecule. It is the kinetic core. And if we look at comparable images, so the catalytic core is seen here from the phosphopeptide activated protein. And here you see the catalytic core from the unliganted PI3 kinase. And the identity of these two is pretty clear. So what we see is the catalytic core, which is stable, but everything else moves around to an extent that we cannot get images. Well, that raises the question whether P85 is still bound to P110. And the answer is yes. We add increasing amounts of a phosphopeptide to activate the protein. And we look at native gel analysis and it stays together. We don't get any dissociation between P85 and P110. And we get very good activation by adding the phosphopeptide. So what should we make out of this? Well, some time ago, Roger Williams did hydrogen exchange of phosphopeptide activated PI3 kinase. And what he found is increased accessibility of the hydrogens in most of the domains of P85. And to put this into a hypothesis, we can look at this last slide. You have a receptor tyrosine kinase here that is active, is phosphorylated. This is the catalytic core, which is stable. And it still contains the ABD, and ISH2 is still bound to the ABD, but everything else is extremely mobile, moves around, binds to the RTK, and facilitates the approach of the catalytic core to the substrate BIP2. This is totally hypothetical, but it is an explanation or interpretation that is in agreement with Roger Williams' data, and that is also in agreement with the data that we have observed. So let me end by making some key points. The inhibitor BIL719 affects Tau signaling, as you would expect, but it doesn't change the basal breast cancer signature induced by the 1047 mutation. And we have identified mutant wild-type specific ligands. These are not inhibitors, they are ligands of PI3 kinase. And that sort of opens the door and says, yes, we can go after mutants specifically. And making mutant-specific inhibitors isn't an unrealistic goal, it's a realistic goal. And then we have started a cryo-EM analysis for the 3 conformational states of PI3 kinase. And there are some functional consequences. Now, before I end, I would like to just give you the story about our cryo-EM. I mentioned to Tom at the beginning that we tried to do cryo-EM at Scripps. We have fantastic cryo-EM teams here, and everybody wants to do cryo-EM. So you can get half an hour, maybe every two weeks to do your work. And it was clear after several years that we wouldn't make any progress that way. We couldn't do trial and error. And I was frustrated, and we had the purified protein and we couldn't do anything with it. So I mentioned that to a friend in Shanghai, and he said, well, I have plenty of instrument time. Why don't you just collaborate? And I said, sure, let's do it. One of my postdocs will come and teach you how to purify the protein, then we go from there. It sounds so easy, but it was very difficult to get NCI to sign off. So I called my grant officer, and she says, well, I can't sign off on this. It has to go to a higher level. So I asked, please move it up to a higher level. And I called back after a month or so because I hadn't heard. And she says, well, the higher level wouldn't sign off. It has to go even higher. So I wait another month. And again, it has to go higher. And it was bumped up all to the top of NCI. And the top of NCI said, no, we can't do this. We cannot approve that collaboration. It has to go to the State Department. So they moved it to the State Department. And I waited weeks again, and then inquired. And fortunately, everybody was sort of smiling and understanding and said, well, it's not yet done. State Department hasn't decided. It wants to move that to the embassy in Beijing. So they did. And after another month or so, the embassy said, okay, you can go ahead. So it's a long story, and it's sort of a funny story and a frustrating story, but it shows the kind of bureaucratic hurdles you have to take to work together with a Chinese lab. But the work has been very rewarding, and they've done a really fantastic job. So here is the acknowledgement. Drugs and growth factors in the PI3 kinase pathway, that's the sole work of Jonathan Hart. The PI3 kinase mutant-specific ligands were done by Su Yang in my lab, who provided purified protein, and Dario Neri and his group at the ETH Zurich. And the cryo-EM in my lab, it's Su Yang and Jonathan Hart, who does most of the computational analysis. And in Shanghai, it's Xiao Lu, a graduate student, and Mingwei Wang, who is the head of the institute, and the Huayang, this is Shanghai Institute of Materia Medica, the Chinese Academy of Sciences. And I thank you for your attention.
Well, thank you so much, Peter. Doing questions by Zoom is always a little bit challenging. Let me start off with a question while other people chime in. The structure that you show for the drug conjugate, the drug bound form, is actually somewhat similar to the 110 beta structures.
Yes, we noticed that, and we made that comparison in our manuscript, and it was, both reviewers requested that we take it out.
Well, you can tell I wasn't one of those reviewers. But it's interesting to me. I mean, 110 beta, people, there's always this question of why all the mutants are in 110 alpha, and in the, there's similarities with the RAF situation, where the RAF mutants are in BRAF, not CRAF. These are very similar, from 30,000 feet situations. Yes. And I wonder if this just represents the fact that 110 beta is better repressed in its basal state.
Yes, yes, it could be. So, of course, we do have plans to look at other isoforms. Gamma has been done, the cryo-EM structure of gamma has been done by John Burke recently. And beta is still up for grabs. What, of course, we are looking for most eagerly is mutant structure. And there's a very simple problem, and that is it's hard to make enough protein or mutant protein because always turned off by the cell, the production of mutant protein.
Yeah.
So we grow it in the presence of Bill 719, and that seems to sort of work. But then that raises the question: if we looked at this on the cryo-EM, is it still Bill 719 bound and has this altered conformation, or will we get the mutant by itself without Bill 719?
Well, actually, I mean, I think it's a wonderful question. I've got one more question, but Luke Cantley has chimed here, and he says, does introduction of activated PI3K into the breast cancer cell line induce changes in DNA methylation that remain stable after addition of the inhibitor? Would adding a degrader to get rid of the mutant PI3K return the transcriptional profile back to normal?
So that was actually refers to an older paper that we published called the butterfly effect in cancer, where we actually showed that if you induce that mutation and look at the transcriptome and then add inhibitors, you don't ever change the transcriptome back to without inhibitor to the normal state. So the mutation does induce changes that are permanent and that are cannot be erased by the inhibitor. So there's clearly epigenetic effect that we don't understand, but that is there.
Well, Joan Berge, I think Joan Berge has asked a related question. Since the PI3K mutant induced the basal state, but a PIK inhibitor didn't block it, do you think that the switch to the basal state is irreversible?
What is the basal state?
I think maybe Joan will answer this, but I think she's referring to transcriptome state that looks like basal breast cancer.
So we look at the transcriptome of wild type versus mutant cells, and can we treat the mutant cells with an inhibitor and get a transcriptome that is like wild type? The answer is no.
Yeah. So is that, have you looked with the Genentech compound, which is roughly speaking degrading the mutant, at least in HER2 positive?
Yeah, we haven't looked at that compound, and it's still, to me, the compound is still somewhat mysterious, and I'm surprised that there are so few papers on it.
Yeah.
Because it's such an interesting and challenging compound.
Well, I mean, the paper describing that compound also brings up a question which is peripherally related to what you've talked about, and that is they suggest that the effects are specific for receptor cells where HER2 is activated, and also for P85 beta, not alpha. You used alpha in your structure. Are you looking at beta?
We're sort of looking at beta, P85 beta, and we find a lot of confusing things that go in the general direction that P85 beta, it tends to be more strongly oncogenic than P85 alpha in various combinations.
Here's a question from Mike Eck. By the way, I'm reading from a list of questions that would go on until It was 10 o'clock in California. So we'll probably not be able to do them all. So Mike X says, hello, Peter, really beautiful work and talk. I share your enthusiasm for mutant selective allosteric inhibitors. With the DEL screening, I wonder if you've thought about a screen counter screen approach with peptide bound versus free to find binders that selectively bind and therefore stabilize the closed inactive state.
Yeah, so we've thought about it, but we haven't done it. And my own situation at the moment in the lab is pretty much that I have decided to close the lab at the end of the year. And I have very few people left. These are all open doors, and I wish people would pick that up. But I don't think I will be able to pursue that myself as much as I would want. I just passed the 90 mark, so I said, no, it's enough.
Yes, when I was reading your CV, frankly, I was shocked to find out that you were born in March of 1932. I would never have guessed that. But, and frankly, I hope you could be persuaded to keep on a little bit longer. This is.
Yeah, I keep on a little bit longer, but I won't have an active lab. And I have so many connections. I want to continue collaborating with the Shanghai group. And they are very eager to do the mutants. And we've already moved into another direction too, where initially we thought, well, let's try a simple way to stabilize the molecule. We'll make some nanobodies. And we chose 3 nanobodies and looked at the structure. And there is some stabilization, but not as much as we had hoped. But we get some increased resolution. Then the question came up, where do those nanobodies bind? And the suggestion was, okay, let's just do crosslinks, and then we digest and do MS, and we know where they bind. Well, that sounded simple, but it turned out not to be that simple. Yes, we could find the nanobody binding sites, but with the crosslinks, we got a lot of more data that also pertain to the structure itself. So we had all these crosslinks that we received, and it was just a big matching game that followed. We assigned the crosslinks, the peptides that were cross-linked, and it helped us identify these remaining 3 domains of P85. That was really identified with the help of crosslinks and digestion and MS. But now we have gotten the entire molecule, the cross-linked non-digested molecule in the cryo-EM. We only have about 3,000 images so far, but it looks like it's going to work. And maybe we get better stabilization with the cross-link.
Interesting. I mean, obviously nanobodies have been extraordinarily useful, but I was just serving on a PhD exam here at Harvard where it really looked like the nanobody structures had actually, the nanobody had distorted the structure of the, in this case, another kinase that was under study.
Yes, and I refer to a non-active conformation that we see. And we see that with 2 nanobodies. We think it could be a nanobody artifact, but the fact that PI3 kinase can assume that conformation means it's not totally impossible for it to also have that conformation spontaneously.
Mhm.
And that structure, I think, will give us interesting binding sites, pockets that have not been seen before.
Well, Peter, frankly, I think this is a good place to thank you so much for this talk.
It was great to be able to do that. And I have looked forward to it ever since you sent me the invitation.
It's just wonderful to have you. And as I said in the beginning, I think you're an inspiration to us all. And I hope you can keep on in some way to keep inspiring the young folks in our community. But again, thank you so much.
Thank you. Thank you very much. Thank you.
Okay, and Claudia, I think we can shut off now.